Hey everybody, we are going to be looking at genetic screening and genetic disorders today and some of the root causes. So genetic screening is a risk assessment for an individual inheriting a certain trait, and you're going to use probability to calculate this risk. There's a general formula that we use where it's R equals F times M times D. So essentially you're calculating the risk, which is R, and you're going to look at the probability that the female carries the gene times the probability that the male carries the gene, and then the risk of the disease under best conditions. So if you have a wife with an albino parent, a husband with no albinism in his pedigree, then what is the risk for having an albino child? Well, because the wife has an albino parent, her probability is 1. She has to have the allele. Because the husband has no family record, we're going to assume he's pretty close to 0. And then since this is a recessive trait, if both were carriers, then there's a 0.25 or 1 out of 4 chance of passing this on. So now when you multiply 1 times 0 times 0.25, because of the 0, you get a risk of 0. However, if this were, let's say, a more severe disorder, we may want to assume, just to be safe, that the husband is the carrier, in which case then we would do 1 times 1 times 0.25, in which case there is now a 1 in 4 chance that any child will be albino. Now, there are other ways to look at the probability of a child having a disorder, and they involve actually testing a fetus. And so in this case, we would, if there is like a carrier recognition, so if you know a parent is a carrier for a certain trait, you would actually be able to complete fetal testing, in which case you would do an amniocentesis or a CVS, chorionic villus sampling, and then there's also newborn screening. So fetal testing, these are biochemical tests and a lot of times chromosomal analyses. And so with an amniocentesis, it happens between 11 and 14 weeks of gestation. Some amniotic fluid is extracted, and then you run biochemical tests in a karyotype. The downside to this is that it does require a decent amount of time to culture the cells. So here you can see that a needle is used to withdraw amniotic fluid, and you separate out the fetal cells, you're able to culture them, grow them in culture, and then several weeks later you can complete a karyotype to check and see that all of the chromosomes are there the way that they're supposed to be. You also have a CVS, or chorionic villus sampling. This is usually administered a little bit earlier, and here you actually extract tissue from chorion, or the placenta. This is a slightly greater risk to the mother, but it doesn't require culture time. And so it's going to do the same thing. In several hours now, you're going to be able to run a karyotype and check on the chromosomes. Again, this is an example of a karyotype, and here we're looking at a female because it's 2 X chromosomes. Now on top of this, most states are going to, I think all states actually, require newborn screening, and so these would be blood tests for recessive conditions that have phenotypes that can be treated to avoid damage. But keep in mind, a genotype can't be changed, so it would just be testing to see if you can treat a phenotype. Required by law in all states, and it's usually anywhere from 1 to 6 conditions that are tested for. So now we're going to take a look at some slightly different diseases, and these are called multifactorial diseases, and this is where you have genetic and environmental factors interact to cause the disease. And so this is something that's becoming much more widely recognized in medicine. A really good example is heart disease. So you can have a genetic predisposition to heart disease, but then there's other things too, like your diet, the amount of exercise you get, and even bacterial infection that also play a role in whether or not an individual will develop heart disease. And now we're going to look at chromosomal alterations. So we talked several units ago about what happens when you have mutations within your DNA. So something like a single point mutation, a change in an amino acid, a change in a DNA nucleotide base that would cause a change in an amino acid. And we talked about how those can range in severity. Well, now we're going to look at something called chromosomal alterations. And this is when you're changing the number or the structure of the chromosome. And so there are several different types of alterations we're going to look at. The first are number alterations. So aneuploidy is when you have too many or too few chromosomes, but it is not a whole set change. So you're talking about having maybe 1 or 2 extra chromosomes, or maybe 1 too few chromosomes. Next we have polyploidy, and this is when you have a change in whole sets of chromosomes. So we know that humans have 2 sets of 23 chromosomes to get our total of 46. So with polyploidy, an example would be if we had an entire new set, and so we would have 3 sets of 23 as opposed just to the 2 sets. Next we have non-disjunction. This is when chromosomes fail to separate during meiosis, and so this is what's going to cause the change in chromosome number. So the result is that two cells have too many or too few chromosomes, so aneuploidy, which we just talked about. So here's a diagram of meiosis. You can have non-disjunction occur during meiosis 1, or it can occur during meiosis 2. If non-disjunction occurs during meiosis 1, then you can see we're going to have 4 cells that all have the wrong number of chromosomes. If, however, non-disjunction occurs during meiosis 2, now we are going to have 2 cells that have the wrong number of chromosomes, but we will still have 2 normal cells that are formed during meiosis if non-disjunction happens during meiosis 2. So again, that's just summarizing what I just said. And so again, aneuploidy is caused by non-disjunction, and it's a failure of a pair of chromosomes to separate during meiosis. And there are 2 general types called monosomy and trisomy. So monosomy, with mono meaning 1, is when you only have 1 copy of a certain chromosome, and then trisomy, with tri being 3, would be when you have an extra copy or a 3rd copy of a certain chromosome. So an example would be Turner syndrome. This is an example of monosomy. So here you have 2n-1 or 45 chromosomes, and you're actually missing the second sex chromosome. So you have an X and then nothing else. So phenotypically, an individual with Turner syndrome would be female, but there would be very poor secondary sexual development. So like all the things that would develop typically around puberty, that's where you're going to see the poor development. Several general characteristics would be short stature, extra skin on the neck, broad chest, usually sterile. And the reason for that is because you have an odd number of chromosomes, so they can't pair up and separate properly. And I just kind of told you this, so that's why Turner syndrome individuals are usually sterile. There are other sex chromosome changes as well. So you have Klinefelter syndrome, metafemale, and then super male. Klinefelter syndrome is going to be a trisomy. So here you have 2n+1, and so you have the genotype of XXY. So even though there are 2 X's, there's also a Y chromosome, and so phenotypically an individual with Klinefelter syndrome is male, even though there's 2 X chromosomes. There is still a Y chromosome as well. And again, you have sexual development is usually poor, but also usually sterile. Here we have Klinefelter syndrome, and we've got 4 copies of the X, but again, we still have that Y chromosome, and so because of that Y chromosome, this individual would phenotypically be male. All right, next we've got metafemale, and this is going to be 2n+1 or 2n+2. So you would have 1 or 2 extra X chromosomes. Because there is no Y chromosome, this is going to be female. Again, poor sexual development and most likely mental impairment. A super male would be 2n+1 or 2n+2, so 1 or 2 extra copies of Y chromosomes. Here's an example. So you got 2 Y chromosomes in that karyotype. Probably the most common trisomy that you hear about is trisomy 21, which is also Down syndrome. And so there's various mental and physical changes that occur with Down syndrome. And one thing that you may notice is that you hear a lot more about trisomy than monosomy. And so one reason that trisomy is more common than monosomy is because any fetus that at conception has a monosomy, so is missing a copy of a chromosome, usually isn't going to be able to survive. And so a fetus can survive with an extra copy of a chromosome, but being hemizygous or having only 1 copy of a certain chromosome is usually fatal. And so that fetus will not develop, and so essentially the woman would have a miscarriage due to the fact that there's not enough information there for the fetus to fully develop. Another question that comes up is, why is trisomy 21 more common in older mothers? And the reason for that is that as maternal age increases, so does the risk of non-disjunction. And trisomy 21 is one of the trisomies where the fetus can survive to birth and beyond, and so it ends up being much more common. Many other trisomies and almost all monosomies will actually stop developing at a certain point. So trisomy 21 is more common than other trisomies in general, and it increases then even more with maternal age because of the increased likelihood of non-disjunction. All right, so now polyploid is when you have a whole extra set. And so instead of being 2N, you would be looking at 3N or 4N, so triploid or tetraploid. So now you're talking about adding a whole other set of chromosomes, or maybe even doubling the number of chromosomes. So if you had 2N, doubling it to 4N. This is not common in animals at all. It's actually usually fatal. However, you do tend to see it in plants. Although in plants, even numbered polyploids are fertile usually, while odd number polyploids are usually sterile. And so the reason for that is because you have that odd number of chromosomes, and so if you have an odd number, they can't pair up properly, and so you end up having issues with meiosis. Okay, so now we're going to look at structural alterations of chromosomes. And so we've talked about deletions and duplications before, but in terms of bases of DNA. And now we're going to look at what happens when you take an entire piece of a chromosome out or duplicate an entire chunk of a chromosome. So a deletion, again, would be if you actually take out a chunk of a chromosome. So you can see here this section labeled D is missing over here. A duplication then would be if you duplicate a section of chromosome. So B and C is highlighted here, and you can see that we've now extended this chromosome because we have duplicated the sections labeled B and C. An inversion would be when you actually flip part of the chromosome. So in this case, the highlighted sections are in the order B, C, D, and now over here it goes A, D, C, B, E. And so we flipped this chunk of the chromosome. And then a translocation involves moving a segment from one chromosome and sort of switching it with a segment from another non-homologous chromosome. So we know that homologous chromosomes switch material fairly frequently during prophase 1 of meiosis 1. A translocation, though, happens when you would say, let's say this is chromosome 1 and this is chromosome 2, and they actually flip-flop a little bit of information. So you can see here the BC from this chromosome and the M and O from the second chromosome actually flip places. Now, if this is reciprocal, meaning that no genetic information was lost, normally this is going to be a regular phenotype because all the genetic information is there. What's going to happen is errors later on, let's say when this individual tries to have babies, then there's going to be an issue with the reciprocal translocation. But other than that, an individual would usually be phenotypically normal. So if genetic information is lost, then you can have some negative effects. If things are flipped, and it actually cuts into the middle of a gene, then you could have a gene's expression kind of being influenced. It may not work properly if you've actually cut a gene, depending on where the segment was taken out or duplicated. And it can also be affected by its location relative to other genes, because you're now switching the order in which you find the gene. Parental imprinting of genes is when gene expression and inheritance depends on which parent passed on the gene, and it's usually caused by different DNA methylations. So imprints are erased in gamete-producing cells and then recoded by the body according to its sex, and so gametes are methylated to code as male or female. We also have extranuclear inheritance, and this would be DNA in organelles such as mitochondria or chloroplast. And so Mendelian inheritance patterns fail. And so you have maternal inheritance of traits where the trait is passed directly from the egg to the offspring, and the example for that is with the mitochondria and the chloroplasts. So chloroplasts give non-green areas in leaves variegation, and this is really common in ornamental plants. There's some more examples that we can talk about later. Mitochondria, again, they have their own DNA, and so you have mitochondrial DNA that's passed down through the mother. All right, that was a lot, and I know that. So here is your lecture question. I want you guys to tell me why you think errors in chromosomes can be worse than errors like point mutations in DNA. So I'm going to ask you guys to think back to some stuff that we learned a while ago in order to answer this question. But please send me a message on the website with your answer. Thanks, guys.